Today we're talking about RNA. RNA is a polymeric molecule with various functions concerning genes. RNA is an acronym for ribonucleic acid and is like DNA and nucleic acid. Well, I guess RNA is not as commonly known as DNA, but at the moment, due to the coronavirus pandemic, we're all talking about RNA. There's even the so-called RNA world hypothesis, which claims that there was a time early in evolution when genetic information was encoded by RNA and not by DNA. This idea came from the fact that RNA has catalytic activities. For example, with the ribosome, which we'll talk about in a minute. However, it's also a fact that RNA is not stable enough and therefore it's not suitable to encode for the complexity of life as we nowadays know it. First, we're going to talk about the three major differences between DNA and RNA. It's called ribonucleic acid and not desoxyribonucleic acid. So we've got a sugar in RNA, which is a ribose. So there's a hydroxyl group in the 2' position, which leads to a chemical instability in comparison to DNA, which is especially true in basic conditions, so RNA is less stable than DNA. The second difference is that RNA is single-stranded, which is quite obvious when you take a look at the two drawings here. It's also possible to have double-stranded RNA, however, as some viruses have a double-stranded RNA genome, for example HIV, which causes AIDS. The third difference is all about the base. Adenine does not pair with thymine as we know it from DNA, but rather with uracil, which is actually an unmethylated form of thymine, so that means that it does not have an additional CH3, so a methyl group, which you can see here. As indicated before, RNA fulfills a huge amount of tasks, but usually you get to know only 3 of them, which are the classes mRNA, tRNA and rRNA. These are the ones that play a role, or the major roles, in protein production, but there are at least 8 other types of RNA which are non-coding, but they're also very important in genes and also their regulation. RNA is physiologically synthesized from DNA via the enzyme RNA polymerase. Then this primary transcript, which is single-stranded, is modified. So at the end there are a few adenines added, which is known as the so-called poly-A tail. Then there's also a guanine added to the 5' end, and its function is actually not known yet, but well, it's plausible to assume that it has something to do with the stability of the mRNA. Yes, that's what we call mRNA. And this mRNA, so the M stands for messenger, this RNA leaves the nucleus and heads to the ribosome, which is the production factory of proteins. The ribosome consists mostly of rRNA, so the R stands for ribosomal, but there are also some proteins in the ribosome. And now, proteins are formed by amino acids. And these amino acids, they have to be transferred to the ribosome first. So that's the task of the tRNA, so the transfer RNA. So, let me summarize. The mRNA goes to the ribosome, which is formed by rRNA, and we need amino acids to form the proteins. They are brought by the tRNAs. That's very important. Interesting is that every protein coding amino acids, and there are 20 or 21, depends on how you define it, has its own tRNA. So, and that's how proteins are built up, which are actually very, very, very important in biology. So this is a very quick overview. RNA is actually very, very complex, and there are so many types of non-coding RNAs, and their tasks are not fully understood. They're also the basis for complex technologies, for example, like CRISPR-Cas9. And so, to sum up, RNA is a complex and really interesting topic. I hope you liked this episode and see you in the next video.